I think there is some lag over here. So what I'm saying is earlier people were first creating the models and then they were trying to identify on what grounds the model has given you the decision, whether the classification or the regression. But now it has changed. People have come up with some models for regression like symbolic regression, which based on the data itself try to create its own equation and then give it the results. So looking at the equation you will get to know that which feature is most important or using which it was done. Similar way, there are some models which in case of medical image is beneficial if you look at this extendable AI concept within the model itself while doing the training itself and instead of waiting for the end result. So within the training itself, if those integrations are done, it becomes easy for you to regulate the loss function using which the weights are updated and your accuracy is improved. Am I clear, Nitish? So if you're going with regression side of medical thing, then that's the case. You go with symbolic regression, that is most useful. Otherwise, you get the integrated features over here for the neural network. Nitish, am I clear?
Yes, sir. Thank you.
Okay.
Sir, there is one question from my side also.
Yeah.
Uh, how do you handle class imbalance in medical image datasets without introducing any biases?
See, first of all, when we are talking about class imbalance, the thing is, I don't want you to increase the data set or I don't want you to reduce some of the samples for a particular case. Like earlier, people were going with smarted technique, Tom Link, or you can say upgraded the features or reducing that samples, upsampling or downsampling. But it's better if you go with the transfer learning, you will not be facing that particular problem, that particular issue. The paper which we published in transaction, that data was highly imbalanced. There were 285,000 images, out of which 175,000 belongs to one class and another class just had around 90,000 images. So it was just the double of the one which was there. So when we went with the transfer learning, it automatically handled it. Going with the distribution was good initially, but now even the distribution cannot, it can reach you, help you to reach about the accuracy of 80 and 90. But now the trend has changed. People are going towards the commercialization. So in order to go towards the commercialization, you will have to use that. So transfer learning is something which I would suggest as of now for this imbalance thing.
Thank you, sir. Thank you so much. We sincerely appreciate, sir, for your valuable contribution today and your very engaging talk. And now I request Dr. Gaurav Kumar, Associate Professor and Head, Department of Biotechnology and Bioengineering, Galgotias University, and co-convener of this FDP, to kindly present the digital memento virtually to our esteemed speaker. Sir, please.
Thank you so much, sir, Rishabh sir, for accepting our invitation to deliver
Thank you,
the
sir.
invited talk. And on the behalf of our Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotia University, as well as our associated and collaborated partner, University of Cyberjaya, Malaysia, I am offering you this digital memento for as a token of gratitude. Kindly accept it, sir.
Thank you so much, sir, and for inviting me for this particular session.
Okay, thank you so much, sir.
Thank you, sir.
Thank you. Thank you so much, Dr. Anjali. Thank you.
Thank you, sir.
I'll take a leave now.
Sure, sir. Good afternoon, all of you. So, let me introduce you to our next speaker, Dr. Krishna Kumar Ojha. Dr. Ojha is an assistant professor in the Department of Bioinformatics at the Central University of South Bihar, Gaya. With over 15 years of teaching and research experience, he has established himself as a leading researcher in bioinformatics, molecular dynamic simulation, high performance computing, computational biology, and database systems. He earned his PhD in bioinformatics from Banaras Hindu University, Varanasi, where his research focused on the computational analysis of replication origins in archaea. Dr. Ojha is a proficient in advanced computational tools, including GROMACS, Autodoc Vina, Python, MATLAB, and high performance computing platforms, and has made significant contributions to computational drug discovery, molecular modeling, genomics, and structural bioinformatics. He has published numerous research articles in high impact international journals, qualified the CSIR net twice, and actively contributes to academic administration, curriculum development, and bioinformatics education. Sir's research continued to bridge computational approaches with biological sciences, driving innovation in drug discovery, precision medicine, and translational bioinformatics. Thank you, sir, for joining us today, and we are looking forward for your expert talk. The virtual platform is yours, sir.
Thank you, Anjali ma'am. Thank you for your
Thank you,
nice
sir.
introduction. Although I am a novice, but anyway, am I audible?
Yes, sir, you are audible, sir.
Okay, so let me share my slide. The slide is visible now?
Yes, sir, your slide is visible.
Okay, okay. Thank you. So I
Sir, one request, sir. You can click that hide button on the bottom of your slide.
okay.
Yeah.
Yes.
Thank you, sir.
Okay. Okay. Thank you for giving me the opportunity to speak on this platform, an international faculty development program on the frontiers in bioinnovations, in bioengineering and biopharmaceuticals, that is jointly organized by Department of Biosciences, Garuda University, and the University of Cyberjaya, Malaysia. Uh, so today, the seminar topic which I am going to present is AI-assisted pedagogy and research paradigm in computational biology. I am a person who was basically trained in biological science, and later I switched to the computational biology. So I tried to cover that what are the challenges a general biologist actually faces when he moves into the computational biology, which is basically interdisciplinary sciences, highly dependent on the mathematics and the computer science. So I have tried to cover the pedagogy approaches of teaching the science, uh, which is now being heavily assisted and reliant on this AI. So these are the outline of my talk. So I will discuss briefly what is the pedagogy of the science, the essential teaching strategy and the technique which we apply in our daily classroom. Now, the paradigm shift of this conventional learning strategy over the AI-assisted learning techniques. What are the opportunities and the challenges which are there in adopting the AI while implementing this in general pedagogy of the science, and how we can apply the various AI tools and techniques in the computational biology. Some of the case study which I have introduced, and their applications and the future perspective. So basically, if we I hope that most of you have heard this term, this pedagogy. So this is a term which is heavily used in educational sciences, education, and uh, this is basically a science and art of teaching. So pedagogy tells us how we can effectively deliver our knowledge to our listener or the students. Now, there is a transition for memorizing of the fact to inquiry-based learning. The pedagogy actually changed from the humanity to the science. In humanity, this is the subject. Mostly we rely on just hearing and memorizing the fact which are being delivered by our classroom teachers and experts. But in science, the paradigm is totally changed. What we are expected that we have to rely on the inquiry-based learning rather than memorizing just fact. It relies on the blend of the cognitive development by implementing the hands-on learning and real-world applicable scientific temper. So it is expected that there will be a lot of practicals, there will be a lot of models which you develop, you will feed the data, you will analyze the result, and whatever outcomes you get, after that you will amend your concept and you will learn the things. Is focuses on the metacognitive cognitions goals. So basically, it's not just rely on just a single principles. It applies the multiple cognitive goals. Like it doesn't apply only memory. It's apply your ability, your other parameters to get the knowledge. And this is a technology enhanced learning. So we this is highly technology dependent. We have the laboratories, we have the different kinds of the experimental tools, and that's why the AI is influencing the pedagogy of the science heavily rather than the other sister subjects like the humanities or the social sciences. Now, the core pillars of these pedagogy of the science are dependent on the three arches. One is the constructivism, the second one is the inquiry-based learning, and the third one is the project-based learning. So in constructivism, what we expect that there should be your own model, your own parameters. There may be your own a way of learning rather than just hearing someone dictations or the theory classes. So what we do, we implement a certain model. Based on that, the student is given different criteria over which the model is fit, and there are outcomes which he analyze themselves, and whatever result he gets, he learn over that result. Then the second core pillar is inquiry. You are not supposed to be a silent listener who is sitting inside the class and just hearing, making notes, and memorizing everything. You should have your own logic, and that should force you to ask questions. So that's how you enhance your learning. So the inquiry is an important and critical pillar of the science pedagogy. And the third one is the project based learning. So what we do, if we have the enough information, theoretical knowledge, and practical exposure, what we do, we try to actually formulate the projects that is based on our theoretical knowledge, and then we try to implement the different techniques, and we hypothesize some models, some hypothesis, and we based on the outcomes which comes from this project, we either accept or reject the hypothesis. So project-based learning is also at the end of the theoretical and the practical knowledge, the core pillar of the science pedagogy. Now, these are the strategy which we implement in our classroom, and this is not the exhaustive list. There are so many other important techniques which we apply in our general classroom. So the important one are the hand-on activities. So that is moving from the theory to practice. So learner work directly with the tools and lab equipments, because the science heavily rely on the practical than just the theory classes. Then concept mapping. So what we do, we use some graphical tools, flowchart to help a student to visually connect new theory to previously learned concepts. So it is verified that the graphs and the figures, the flowcharts, they are more penetrative to our mind rather than the theories. And then peer-led team learning. So it facilitates collaborative group formation and solving a complex problem by subdividing to individual group member. So we make some group and we give certain exercise, and we accept that everyone will get even opportunity to contribute according to their ability. So that improve both the social as well as the analytical students, analytical skill of the student. Now, the question is how AI can strengthen the pedagogy of this science. What we done earlier, that we have the classroom, we have the model, we have the interactive flow charts and all those things, and we were using conventionally in our classroom to teach the science. Now, in the era of the computer and advent of this AI technology, we do have much vast scope in enhancing the pedagogy of the science. So now we can implement the interactive simulations. We can adopt the personalized pathway to focus on those students who are not the easy learner one. We have different kinds of the audio visual visualization techniques through which we can use the several either VR or the augmented realities to showcase the our data. And then the creative communication. So we do have those students who are not the native learner of the English, or those students who are in a tribal area who have not been given actually exposure to learn the English. There are translator which are available, which in minutes, although they are not 100% correct, but they can be taken care of, and they can help the student to translate in their conventional vernacular languages. So using these technologies, we can enhance our pedagogical aspect of explaining the science to our student. The interactive simulations, we do have so many simulations. Without actually doing the actual experiment, we can adopt them. That cut both the cost as well as the time of the actual experiments, and the student can learn in an effective way, cost-effective way. Now, I think enough exposure has been given by the previous, expert on the AI. But because I am going to deal with this, AI in the pedagogy of the science, so I will give you a brief introduction. So, artificial intelligence is basically a branch of computer science which uses building a smart machine capable of performing tasks that are typically require the human intelligence. So in other words, we can say when the machine take the logical decisions like the human, then it becomes the a smart machine, and that smart machine is using artificial intelligence in its backend. So instead of following the rigid pre-written codes, the AI system analyze massive data set to recognize the patterns of complex problem and make autonomous decisions. So this is very important aspects of the AI, that any AI requires a training. Okay, so AI is a bigger umbrella, which I will discuss in later slides. And the basic component is the machine learning. How machine learn? So we have the massive data set, and that is used to train our system, and taking the training basically, which is nothing but identifying certain patterns and making the internal logic rules. Machine are able to do and take the logical decisions, and that's how this AI work. So this is the umbrella relationship, which explains the relationship of AI, machine learning, deep learning, and the large language model. So the deep learning and LLM, which is basically a product of this deep learning, these are the very new aspect of this AI. Machine learning is not so new. That is, we are using machine learning, I think, from the 15 gigahertz and older. And so this machine learning is basically a subset of AI that use algorithm that learn the patterns and make predictions from the data. How accurate machine learning will be depends on that what is the type of data, the amount of data, and the algorithms which is used to train and make the machine model. So that depends on the various factors. Now a new type of the learning is deep learning, where we use the neural network, which are basically a mimicking the neural network, actually mimicking our own brain neuron. So it's not just linear learning. There are various layers of these neurons and neural layers which are being trained, and there are various kinds of the parameters which can be changed to train our model. So this is more specialized. That's why it is known as the deep learning. It can gain much insight. It can gain the much insight from the data as the conventional machine learning. And the last one is the large language model. So the advanced applications of the deep learning, like the Chat GPT, or Google Gemini, or the Microsoft Copilot, all are the product of these large language model. So how they learn? Actually, we feed themselves the large chunks of these text, and the text may be the sample text, the text from the books, and the entire vocabulary which is available on the web. And what they do, they actually make the relationship with the word. So how they learn, actually they measure that what are the probability or likelihood that how the two words come together, so how they are joined together. Okay, so that's how they make their rules. And using this large language model, they act like a co-pilot, which you can use like an agent to ask the question, to compose paragraph, to write and speak on some topic. So it has actually revolutionized this AI age. Now, why this computational biology is a great hub in implementing this AI? So actually, we are using a precedence a dividend in computational biology for implementing the AI, because we are already using the computer to store, analyze, and get the meaningful knowledge which is hidden in the vast biological data set. So we are already using the computers and the different statistical techniques to gain meaningful insight data from the large chunk of the biological data set. The second thing is, I have already mentioned that how competent will be the AI model, that depends on how massive data it has been trained. So luckily, the biology, in biology, we do have the largest set of the data sections. Most of the biological data sets are extremely large, in petabyte size. And it's easy to train the AI agents on the biological data to get the meaningful informations, which is not conventionally possible without the AI. Now, we have, if we want to find the hidden rules, which is embedded inside the expression data of the gene. So we in human body there are thousands of genes which express themselves at various at various point of time, or there are several domains of the genes which express themselves, which are triggered by the various signals pathway inside the cell. So we can decode the hidden rule, that what are the key parameters which trigger them to switch on and switch off. The AI has accelerating the discovery of the new medicines, new lead compound by drastic cut down in the times which has been taken by these screening of the thousands of the molecules in the lab, and then getting the lead compounds, and then further type 1 and type 2 drug discovery stages. We can implement the AI for the pattern recognitions, which extract the hidden signature from the massive raw data sets. Like this technique we can use in getting the biomedical data set. Like suppose if we are getting the CT scans of a tumor patient, and if we will divide the images in pixel-wise manner, which can't be actually recognized by normal human eyes, then we can get the features which are associated with the tumor versus the healthy cells, and we can easily identify the tumor. Then we can use this AI technique in prediction and cataloging of these different kinds of these cellular networks and their behavior. So these are the core area of the computational biology. This is also not the exhaustive list, but we can actually divide the total computational biology in this major 5 subdivision. These are the core omics technology, which has been further divided into transcriptomics, genomics, proteomics. Then the structural biology, which are the backbone of these computational biology. Then the subdomains of this is computer-aided drug designing, in which we do screen the millions of the molecules which is available in silico, and then we apply the computational tool to filter them out using the virtual screening technique. And then we do have the actually method which is known as the docking, through which we can assess the binding affinity of these molecule with these protein. And then we can do other parameter to identify the lead molecule among the millions of the molecules which is available. Then another aspects of this computational biology is molecular phylogeny, in which we try to infer the relationship among the organism based on the molecular data. So we have the molecular data in the form of the DNA, RNA, or protein sequences, and then we try to infer that how these organisms are related with each other. There are various kinds of the AI technique which we can use to enhance in deciphering the relationship, because this is highly space reliable. So time and space complexity of this molecular phylogeny experiments are very high, because when the sequences become large, or there are multiple organism which is available, like if we are trying to deduce the root of life, then the total number of the organism as well as the sequence which we have to investigate, they become very large, and the time complexity and space complexity are increased dramatically. So that's how we use this AI to reduce this time and space complexity in molecular phylogenetic experiments. And then computational neuroscience. This is a recent field in which we apply the computational technique to identify the various kinds of the hidden patterns which is hidden inside these MRI slides. Now, we apply various kinds of the AI agent in these 5 domains, starting from the omics technology to the computational neuroscience, which I have described earlier. So in genomics, we can use this Google DeepMind, which has a series of tools. So it has a series of tools which help us to, parse the literature, the whole literatures which is available on the PubMed. And then not just the simple literature source, it can use various kinds of the filters to get the meaningful insight. After that, it can integrate those filter to accumulate a hypothesis generation and a computational discovery of the problem which we can use for our research. The AI can use in DNA sequencing accuracy. So in high throughput sequencing experiment, the rate at which the DNA are sequenced is very high. But the same way, the inaccuracies which are being accumulated over the sequencing is high. So we can use this AI to minimize this inaccuracies. We can use AI in filtering the noise and error in raw genetic sequence data. Tools like this, Google Deep Variant read the genomic sequence like the images, which uses the convolutional neural network to accurately identify the genetic mutations. And using this, we can find the single nucleotide polymorphism, which is responsible for the allelic variant or which are responsible for certain genetic rare disease. We can use this var chat to compare the human sequence genome to health data set to predict the medical risk. So now the sequencing become very cheap. The entire human genome can be sequenced, within rupees, 1 lakh. And later, using the whole sequence data, we can give to the var chat, and it will compare with the health data set and the online Mendelian inheritance of the man, and it can predict the our medical risk. So how much prone we are to develop certain kind of the cancer, how much prone we are to develop the type 2 diabetes, we can predict in a very accurate manner. So it's not only the coding part where we apply these, AI, but we know that 99% of our genome is doesn't code anything, but it play a regulatory role. So genomic foundations like the Informer and Evo, they learn the structural grammar of the DNA. And because the non-coding region are available in very large amounts, so we can enforce these Informer and Ego to learn from this non-coding region and predict that how much mutation in non-coding region affect the gene expression, because it is established that most of these non-coding region responsible for they are responsible for the regulatory role in the expression of the genes. The same way, the co-scientist is a multi-agent AI system that iteratively generates the debates and evolve novel hypotheses for the complex scientific problem. So we apply all this tool to learn the patterns which is hidden inside this non-coding reason, and then apply this rule to infer the regulatory role which are being played by that segment of the DNA to the expression of the gene. So the same way, the transcriptomic, that is basically the branch of deciphering RNA and the gene expression. So it understands which gene are active, dormant, are responding to the environmental changes. So it's not only the internal mechanism through which the genes are switch on, switch off. There are various kinds of the environmental factors which trigger the gene to switch on and switch off. So dynamic mapping of these RNA taking reveals which genes are turned on and off under a specific conditions. And the deep learning model like the Google Deep Network classify the disease subtype based on the RNA expression signature. So the pinpoint precise biomarker for early cancer detections and other things can be identified using these techniques. So epigenomics, obviously, these are the branch in which we study that there are modulations beyond the genetic control, and these modulations are the environmental factors which changing the gene activity without altering the DNA sequences. And the CNN network can predict the cancer-associated DNA methylations and the chromatin layout, which is responsible for the onset of a cancer. And it tracks early marker for chronic disease and aging patterns. So epigenetic clock are AI algorithm that estimate our biological age by reading the DNA changes. So epigenetic clock, if we have the genetic sequences, we can give them to this epigenetic clock AI, and that will tell us by just marking the DNA methylation patterns that what should be our biological age. Now the structural biology, which is very important aspects in the computational biology and bioinformatics, analysis of the sequence is not that much challenging as compared to the solving the structure of the protein. So the challenge of finding the protein all known protein structure, when there was no AI and the computational model, it was very tedious job to inferring the 3D structure of the protein. We conventionally apply the X-ray or NMR or cryo-electron microscopy to know the structure of the protein, but that is cumbersome. Not just taking the structure, even to crystallize the protein or preparing the solution structure to get the NMR spectra, that is very tedious job, and you can't guarantee that how much time you will take to get the protein structure. Albeit getting the protein structure is not always satisfiable, because most of the time we get the structure, but the important part of the proteins get missed. Okay, so that happens generally in the X-ray crystallographic structure, that the regions which are highly mobile, they are left out. We don't get the clear diffraction patterns of the electron, and that's why they are missing in the original crystal structure. So to actually solve this problem, we use the AI, and this AI has revolutionized. We should thank to the Google that has given us AlphaFold in 2020, and that has actually changes the whole paradigm of this computer protein structure prediction. So there are several versions of this AlphaFold 1, 2, and presently AlphaFold 3. So over 100 millions of the protein structure made openly available through this alpha fold 3 model. And you can see this modeling accuracy. So in this graph, you can visualize the CASP is critical assessment of these structural proteins. So this is an agency which actually measure the accuracy of the prediction of a particular protein. So the computational model is used to predict the structure of the protein, and this is the global distant score, which measure that how accurate your protein structure is. So that is similar to the RMSD, but there is a minor difference between this GDT over this RMSD. Actually, in RMSD, what happens that no matter if you are getting a good structure for a particular reason, but if you are getting a bad structure for a small subset of the protein, the whole score get affected. But this GDT is not like that. That has minimized the effect of this low score boundary. So this AlphaFold, you can see that the prediction accuracy has surpassed the 90%. That is just the data from 2006 to 2020. The AlphaFold 3 has surpassed the prediction accuracy up to 92%. So that is very much accurate. Otherwise, the other methods which we previously adopt, like the homology modeling or the ab initio modeling, they have so many problems. If you are doing the ab initio model, so it can't model very a large protein. In homology modeling, we can model the protein, but the accuracy depend on that how much reliable template which is available similar to our model protein. So if our protein is very new one for which there is no template, then the accuracy drastically reduces. There is another important aspects of modeling these modeling the protein using this AlphaFold. Most of the proteins which structure has been deduced from the experimental technique, like these NMR or the X-ray crystals, they are being co-bind with some other cofactors, metals, ions, or several other things, ligands, small ligands. But in AlphaFold, you just have to give the amino acid sequences, and it predict the structure, give you the structure of the proteins with accuracy very similar to the experimental one. Now, the important aspects of this structural biology is AI-driven drug discovery. So we know that the traditional approaches of discovering a new drug for a disease is very cumbersome. It takes around 20 years for a new drug to come into the market. But the AI-driven, computational approaches has cut this line from up to 5 to 7 years and more. So old way of using only this computational way to finding the biological root cause of the disease took up to 5 to 7 years. But the predictive algorithm process, biomedical text mining, which we can do using this generative AI knowledge graph, and further the virtual screening of the millions of the molecule from the zinc or the Campbell, and apply these AlphaFold and docking and machine learning classifier for admit predictions, we can drastically cut down the time which has which is taking from the virtual screening of the large data set up to finding a novel lead into some mere month. Sometime that can be even sequence to week. So that are the breakthrough in AI-driven drug discovery. That it's not only the identification of the lead molecules. There are various barrier which you have to cross in drug discovery, and that is the drug trials. So in drug trials, there are several levels, 1, 2, 3 level. But now with AI, we can do these digital trials. We simulate the drug to target binding affinity using the physical informed AI models. We can use this generative chemistry, which suggests the thousands of the same molecular compound that are tailored for binding a particular site. Model like this ChemBot learn the grammar of the chemistry to predict the physical property of the molecules which are essential to bind in a particular protein. So that drastically cut down the time of this virtual screening of millions of the compounds. That increases the efficiency by minimizing the manual, expensive, and trial and error chemistry in the physical labs. And these generative adversarial network, they reinforce the learning loop to generate the novel chemical architecture. So it changes the various kinds of the different subtype molecular group which can be attached with the ligand to effectively more binding with this protein. Biomedical imaging and facial biology also can be revolutionized by using this AI. So we know that extracting pixel into cellular insight through this CT scan or the MRI sees, and CNN model evaluate histopathology, tissue slides, and high resolution cell microscopy images. And they chunk down these pixels further into the matrices, and they convert these pixels into the numbers, and it's very easy to compare the number than just compare the images. So spot microscale tumor boundaries that might be missed by the human eyes can easily be identified using this generative adversarial network model, AI agents. And they provide diagnostic quality images while exposing patient up to 7% less radiation doses. So this is also a very important aspect. Whenever we do the CT scan, so that depends on that how much layer of scans you are using, and the durations for which the X-ray are being given for that patient. So CT scan are roughly 10 or more time higher as risky as compared to the X-ray, and more doses you get than the conventional X-ray. So using this GAN agent, you can easily reduce the total amount of the doses which is required to get the high contrast images from the CT scan data. The same way AI can be used in MRI to reduce the time that is required to get the resonation data of the patient from 45 minutes up to 10 minutes per patient. So that is a great achievement. We know that the MRI machines are very costly. They require a high sophisticated environment as well as the expertise to run them. And in a single day, you can't do more than 10 MRI. But using the AI, because the time which is required to get this, take this spectra has been reduced 10 times. You can take up to 40 to 50 patient MRI in a day. And now the AI has revolutionized in reading the scientific literature at the scale. Notebook ML, which has been developed by this Google, They can be used to comprehensive literature search to find the relevant paper, structure, result, and the data table. It not only search the relevant literatures through your search term, but it creates the table, it creates the graph, and through the data it can give the hidden patterns also. So it create a polished presentation outline to complete with the key checking points and supporting evidence by just uploading the source text. And millions of the biomedical papers are can be analyzed simultaneously in a minute, which is outstripping human reading capacity. And the large language model like the BioBert and MedGemma, they scan database like this PubMed. And the benefit is that it's extract the implicit relationship between the drug compound and side effect buried across the diverse paper. So the gist is that you can analyze and get not only the data, but you can use those. You can store the data, you can mine the data, store the data, analyze the data, and get the meaningful insight from just using this Google Notebook LM, which is not possible by the conventional platform like the PubMed. Okay, so we can apply the just the filters to filter out the relevant searches, but we can't extract the data which is written inside the paper, which are meaningful for your research term. Okay, so this is an added advantage of using the Notebook LM. So these are some of the essential tool which I have used, or which I assume that you should also try to use if you are doing bioinformatics, or doing analysis of the biological data using the computers. The first one is AlphaFold 3. It is just the advanced version of the AlphaFold 1 and 2. So it creates the 3D structure of the protein, DNA, RNA, molecule, chemical ligands, and the chemical modification directly from the sequence input. So you don't have to do anything. You just get the sequence either from a UniProt consortium, or from any other source, and just you give the input and it will create the 3D structure. It's not only the create the 3D structure, it has also the component through which you can identify the different domains. You can identify the conserved region motifs, and you can even classify the protein into that from which family the protein belongs to. Then this Rosetta Fold, which is the advanced version of the Rosetta, which is used as an ab initio model agent, which is a all model, all atom model modeler. And this is open source tool which accurately track the structural folding across the protein, nucleic acid, and small molecular interfaces. So this Rosetta Fold is an open source, but this ESM Fold is not an open source platform. But you can use it for academic purposes. Then the DeepDoc. So this is a AI-enabled diffusion-based generative model that map the molecular dockering configuration of a ligand with the target. Deep variant is used in genomics and transcriptomics, in whole exome data analysis. This is an open source analysis pipeline, which is based on the convolution neural network to accurately identify the genetic variant and SNP from the raw DNA sequencing. So this is very useful. You don't have to write the massive code in R to get these SNVs and intels. You don't have to parse your data, raw data. You don't have to clean it, but this D-variant will do all these things for you. So if you are novice and you doesn't know the jargons of these, step of the in the transcriptomics and genomics, you can easily get your result by just using this D-variant. This Science GTP is a generative pre-trained foundation of the cell type annotation, which uses the multi-omics integration data and genetic perturbation tracking. So it can get the data from the single cell RNA-seq, and it can predict how the group of genes switch on or switch off. Upstreaming and downstreaming genes can easily be identified using this as GPT. Then there are some analytical platform like this NIME for visual code-free data analysis workflow, and there are various kinds of the pipeline maker like this Nextflow, SnakeMake, and the Docker, which we usually use when we have to automate the whole process. So suppose in drug discovery, you have to first identify the target, then you have to identify the active site in the target, then you have to screen out millions of the molecule against those target active site, again you have to do the toxicity analysis. So all this process need to be tied together, and you can make a pipeline, so the user don't have to do all this process individually. You just start one process, and automatically all those output from the one process become the input of the next stage process. And for this we use these pipelines, and the next flows. This Docker is an excellent one which we use in our daily life. So they are very helpful. Now we will discuss what are the major challenges which we face when a biologist enter into the computer science domain or the mathematical domain. So life sciences student possess biological intuition, but they're paralyzed by the programming syntax, and the computer science student build this obviously vice versa flawless code, but they are not very well understand the structural biological system. So that impose a imposter syndrome, which is basically that when you feel that most of the people who are sitting around you, they understand some trivial problem, and if you are not exposed to that problem, you fear to ask technical question. Okay, so this is a common problem which is faced by the biological foundation student in computational class. Steep learning curve in the computational biology due to fundamental shift from the descriptive science to quantitative analysis. Most of the students are not exposed to this command line, programming or command line environment, which is the basic one to analyze the large chunk of the data. Most of the time, most of the tools, they doesn't have this GUI in their open source domain. So you have to run the command line, and actually the biologist actually they fear of using these command line interfaces. So that create a problem of these curriculum disconnect. Curriculum disconnect, it means that you have a particular fear because you are not exposed what you are going to learn. Like if we have been trained in biology from class 9 to graduation, and suddenly we use the Linux machine, we have a fear of, and we become a curriculum disconnect problem. Now this educational the NEP 2020 have tried to eliminate such barriers. So this NEP 2020 has the provision that the student are not being segregated, or they are not being clubbed for a particular branch, but they should be encouraged to take subject of their own choice. So there is no boundary. A mathematical student can learn biology, and a biology student can learn the mathematics easily if he has a choice, and if he can make a pace with the other student. And the drive lab are being turned into a discovery space where the student are oriented by exposed to different kinds of the models and the different kinds of the simulation experiment. So the NEP prepare Indian science, technology, engineering, and mathematics graduate to transition from the technology consumer to algorithm creator by fostering the cross-disciplinary research and collaboration between the science, tech, humanities, and the art. Another challenge which we face is cognitive leapfrog effect. And what is cognitive leapfrog effect? That most of the Indians are limited to lack of access of the local high performance computing. So basically, what happens that when you are addicted to a very low level of the technology sphere, and suddenly get a very high level of technology paradigm, there becomes a lag of assessing and understanding the core principle of the technology. So this AI is democratizing the access by empowering the tier 2 and tier 3 institution, which runs the cloud-based biological modeling, and refocusing student attention away from the technical execution and biological meaning. You student, there are some drawback of using the AI also, like a student use a large language model to generate error-free codes. This is the general problem which we face in our classroom. They use the co-pilot and robot to get their assignment done, and they can generate the script within a minute without going into the jargons of this code and the logic. We do have to adopt this strategy like the oral defense and supervised work to evaluate the genuine human judgment, okay, rather than rely on their actual competencies. The another aspect is blind trust in black box model. So most of the model which we use, most of the tools which you use in the biology, they will give the result whatever data you will feed it, because most of the machine are now using these AI. So what happens, they are now no longer menu driven. So all processes are almost automated. You just have to upload the data and you will get the result, but you do not know that how reliable your score is. Okay, so we should aware of that. We should not blindly faith on this black box model which is generated by this AI. And the another important aspect is plagiarism. So in dry lab, because most of these traditional assignments are using AI tool, multipass evaluation technique are being used by large language model. There are various kinds of the paragraph phrasal is available. So if you copy something and you just rephrasing you do, it's very hard for the recent one plagiarism checking software like the Turnitin. So they bypass all these tools. So now the AI has created excellent detector like the size space, which uses the mathematical burst line and linguistic perspectivity for flag a structural text manipulation. So you can easily identify the text which has been not generated by this Turnitin using this size space. You should use this. Now, the threat of academic shrinkage. What is academic shrinkage? When we have the technology, we overuse it rather than actually doing our own logical and cognitive ability, involving cognitive ability. So that is responsible for academic shrinkage. So there is a decline in the deep critical reading skill among the early career life science researcher, and that is also alarming. So we have to assess the process and not the product, and adopt inquiry based learning model to overcome this academic shrinkage. Now I have already described that AI opportunity in the computational biology. So we have a now personalized AI agent like the Copilot, which help us to create compiler error free messages and code, and that is transitioning a teacher role from simple script debugger to a high level research guide if you use, but it lower the failure rate in introductory bioinformatics modules. This AI can give you ability to create the custom synthetic databases. So if you want to if you have a target and if you want to design a database using the pharmacophore model for a given target, you can create millions of the compound using the AI. Like this notebook LM, it create, analyze the generate of report of the simulated model data of this then demystifying complex command line toolkit. So traditional bioinformatics pipeline depend on the complex bash command, which we use in Linux, Docker setup and next flow script. But AI tool that convert the plain natural language instruction into the functional optimized terminal commands. So they can actually help us to overcome this uh, the fear to use these command line interfaces. Then this is the another aspect. Most of the students which come from the rural areas, they do have problem in understanding the English language. So Anuvandini AI that has been developed with collaboration with the CDAC, it can translate the English language into 44 regional language, and you can easily get knowledge of that. I think I am out of time, so I will just go to the this is simulated molecular docking sandboxes, which are AI-driven virtual lab computer-aided designing tool. So that cut down the conventional virtual screening experiments. The same way this notebook LM can be used for literature synthesis from the PubMed. So it actually extract the data from the PubMed PDF documents, and it can analyze them. The augmented classroom blueprint, a comprehensive look how AI fit into. So actually, we don't have to fear about this and completely block this AI agent from our classroom. We should use an augmented way to to get the benefit of this AI tool by active teaching design. We choose actually employ the interactive oral examination rather than taking the assignment, from ensuring deep subject ownership. And we should implement this 5A model. This is a word in AI, that is engage, explore, explain, elaborate, and evaluate, to get the maximum benefit from the AI in biology. Focus in Yeah, we can use in this now ethical protocols, which is very important by using the AI. So creating explicit institutional guideline that separate the valid AI assistant from academic dishonesty. We should foster the culture of honesty, transparency, and accountability in scientific writing. So this is our duty because no one can compel us. The tools doesn't have the criteria which format us to include the content or exclude it. Now there are vast difference in the digital access, particularly in India, in the urban as well as the rural universities. So we should design the lightweight and mobile-friendly AI application that acquire the minimal data bandwidth. So this is a bottleneck while using most of the AI tools. They require high bandwidth of the data to access them and analyze the result. So this is also a challenge. And this is the final summary. So transitioning biology from a perspective science to a predictive engineering discipline, avoid from manual data pipeline to automated AI driven discovery loops, and stress on the cognitive strict code transparency to open data validations, obviously, so you don't have to blindly faith on this black box output. And this AI will not replace the computational biologist, obviously, no matter how efficient the AI is, but computational biologist who use AI will replace the one who doesn't. Okay, so this is all I have. Thank you for your patience and time. So if you have, I think I am out of time, so if you have any short
Yes, sir.
question you may ask. Yeah.
Thank you, sir, for this very engaging talk. There is one question in the chat box. I
Yeah.
can read it. Can we do molecular docking with multi-drug compound or combined drug with multi-target or various disease targets at the same time?
Yes. We can do it. I think we should take the help of this notebook LM. It has a component of doing this, and this AI docker. Yeah, this is a specific tool which can be used in docking millions of the compounds. This has also the capability of this pharmacophore modeling, and it can generate a vast library of the molecules for a given target, so we can use that. I think
Thank you, sir, for the wonderful answer. Is there any more question from the audience? We are looking forward to the participants' queries. If it is that, you can put it in the chat box also, and you can raise your hands in the raise hand features. Suyog Kiran Harugade has raised hand. Do you have any question? Go unmute.
Uh, Sir, I have a question. While checking the plagiarism of the given data, it will be what app we use or AI tool we can use to make that easier or simpler to check the plagiarism is okay or not.
No, so if you are using the conventional tool like the Turnitin, so you can do that. Even the recent version of the Turnitin, I have experienced that it is able to make it out those sections which you have rephrased. Okay, so you can do that, and using this Google Notebook LM, you can easily identifies those sections which has been changed using the various paraphrasing tools. So you can do that. Google Notebook LM is a very powerful tool, and using that you can do various kinds of these activities. But if you have to just checking the conventional paraphrasing, you can use this Turnitin.
Thank you, sir. Thank you very much, Dr. Ojha, for your excellent and highly informative presentation. We sincerely appreciate your valuable contribution to this FTP program. Now, I kindly request our Associate Professor and Head of the Department, Dr. Gaurav Kumar, to kindly present you with the digital memento virtually. Sir, please.
Thank you, Anjali. Thank you, sir, for accepting our invitation, and I am highly grateful to Manish Tripathi, sir, also that who has invited you. On the behalf of Department of Biotechnology and Bioengineering, School of Bioscience and Technology, Galgotia University, and our collaborator, University of Cyberjaya, Malaysia, we are glad and honored to offer you this digital memento, sir. So kindly accept it. And thank you, sir, for delivering such an insightful talk.
Thank you. I'm privileged to have got this opportunity to present this lecture before you. Thank you, sir.
Thank you so much, sir.
Thank you, sir. So we will proceed with the next speaker of the session, who is Dr. Prerna Singh. Our final speaker for today is Dr. Prerna Singh, Senior Project Executive Officer at the Vadhwani Innovation Network, Center of Excellence, IIT Kanpur. Dr. Singh obtained her PhD in biotechnology from BHU and completed her postdoctoral research at IIT Kanpur. She worked there extensively on translational biomaterials, tissue engineering, chronic wound healing, and regenerative medicine. Ma'am has also served as a visiting researcher at Aalto University, Finland, working on 3D printed biomaterials and polymeric scaffolds for biomedical applications. Her research interests include biomaterials, regenerative medicine, tissue engineering, polymer modification, 3D printing, phytomedicine, and translational biomedical research. We are honored to welcome you, Dr. Prerna Singh.
Thank you. Thank
And
you
we
so much,
invite-
Dr. Anjali, for the kind introduction. And
Thank
I'm
you.
very grateful to be here on this platform today. Thank you for inviting me and having me. And could I please share my screen now?
Yes, ma'am. Sure.
So is it visible and in full screen? Hello?
Not yet, ma'am. Yeah. Yes.
Yeah. So thank you once again, and I congratulate all the organizers for putting up such a great event of faculty development program, and for having me for the invited lecture. So my research expertise lies in soft tissue engineering. I am a skin tissue engineer, and I work with biomaterials and strategies for treating complex wound conditions. But not only that, I am also associated with this Vadhwani Innovation Network Center of Excellence, and it is a center recently which has come up in IIT Kanpur. And although in other places it has been there for quite a long time, but it has come recently in IIT Kanpur, and these centers are specifically dedicated towards translational research. So that is something I'll be also talking about and highlighting in my talk for today. That is advanced tissue engineering approaches for wound healing, from research innovation to product translation. So, before starting this lecture, I will just start by asking a simple question, that how many of you had recently experienced a small cut or wound? We generally face it in our day-to-day life. So the answer will be yes. Many will answer yes. And what did we do about it? So the answer will be nothing. So if you have a small cut or something, mostly you will put a band-aid if required. Otherwise, we just leave it like that, and the the wound gets healed on its own. So that is the beauty of human body, that most of the conditions, if they are not chronic, if there's not any disease condition, it is able to take care of itself. But when the condition becomes chronic, or the trauma is very big, so then intervention is needed. So wound healing occurs by repair and regeneration. So repair is something you can associate it with the tube of a cycle tire, and it gets punctured. So what do? We put a patch over there, right? Then it goes back to functioning, functioning normal, and you can drive the cycle and everything, but you can still see that patch. So when we talk about wound healing, you can associate repair with that. That the tissue when gets healed, also, although it works properly, but it doesn't, come back to its original form. So that is repair. And being a tissue engineer, we always aim towards regeneration. That the newly formed tissue should be as should mimic as closely as possible the original tissue. So this is what we are aiming in tissue engineering and regenerative medicine. Since my area is wound healing, I'll be focusing more towards that. So there are two types of healing. First is primary intention and secondary intention. So in primary intention, the when the surgeons and doctors put a very fine give a very fine incision, so the skin surface can be approximated and you can suture it, so the scar formation is very, very fine. Next is secondary intention, which happens in most of the cases. So whether trauma is very big, tissue loss is very big, and some accidental case or some due to some disease condition, there's a big loss of tissue that happens in secondary intention. So the surface of the skin is irregular. You cannot approximate it, and big scar formation is there. So mostly interventions are needed for secondary intention, and if diseases are there and some bacterial infections are there, so we need different strategies to treat such wounds. So I'll just give a little background of the stages of wound healing. A normal wound takes around, if it is not complex or chronic, it takes around 2 to 3 weeks to get healed. So healing stages occur. Four primary stages are there. So hemostasis, inflammation, proliferation, and remodeling. So each of the stage is very, has a huge complex mechanism on its own. We will not discuss it, in today's session, because it will be another very big topic to cover. But what happens is hemostasis is the first step. So body's automatic response is to stop the blood. As soon as the trauma happens and any wound or cut is there, the body will try to stop the blood which is coming out of the wound. So that happens in hemostasis, and it happens within seconds to few hours of the trauma. Second is inflammation. Now the wound site is infiltrated with debris, necrotic tissue, and all other debris. All these things are there. So the body needs to clear it out. You need to clear that space for new cells, new growth factors, new signaling factors to come in. So that happens in case of inflammation. That take few days. This inflammatory phase. Now comes the proliferation. Here main healing starts to occur. So the new cells are coming in, and new tissue formation is happening. New vasculature is building. So in the proliferative phase, we have all the generation of the new tissue. Now what happens in remodeling? This is the last stage, and this can take up to few years also. So in remodeling, the body tries to align all the newly formed tissue and all the cells in the original manner. So remodeling is where the fixing of all the and aligning of all the newly formed tissue happens. So this was a little background about wound healing, and all these stages are associated with different factors, different inflammatory cells, different kind of signals. So when we start thinking about the strategies to develop a biomaterial or develop a scaffold for wound healing, we have to think on these lines. that where the trauma is really which stage is getting affected, what is the condition of the wound, which yeah, stage should be targeted. So all these things should be kept in mind. So, a chronic wound is often associated with several hallmarks. What we talked about was a normal wound healing, how a normal wound healing happens in normal stages. But during chronic wounds, there are certain hallmarks that are associated. That now these can happen in disease condition, trauma conditions, or some infection conditions. So, oxidative stress is one of that condition where the reactive oxygen species are produced in a very large quantity, and antioxidants are not able to overcome them. Then we have the hypoxic conditions due to poor vasculature, and nerve endings are also damaged. So oxygen supply is very less at the wound site. Poor vasculature, again, I already described that due to trauma or something, the vasculature may get disturbed. Now, bacterial manifestation, if the wound is open and we do not take care of it, if it is lying open, then bacteria can come and manifest, and then again sort of a prolonged inflammatory phase, and all these things can happen. So these are some of the certain hallmarks associated with a chronic wound, and to overcome these, we need to have a combinatorial approach. See, all these conditions cannot be tackled with a single strategy. You cannot take a single molecule and imagine that all these things will take care it can take care of all these things. So we need to have a combinatorial approach. Now, in regenerative medicine, we always talk about scaffolds. Scaffolds can be imagined as a matrix which are placed at the wound site, and they provide support to the wound conditions. They have we can incorporate various growth factors, various signaling factors to them, and they help the cell to new cells to infiltrate, new vasculature to develop, and aid in the conditions where normally wound will not heal on its own. So, talking about growth factors, and for oxidative stress, we can have some antioxidant molecules, and for signaling, we can have some signaling molecules such as nitric oxide is very famous in wound healing materials, since it has a plethora of properties associated and beneficial for wound healing. Then we can have some antibacterial properties, some oxygen releasing properties. So, giving an overview of my work, these are some of the strategies which I applied during my research. So, first one is functionalized scaffolds, and all these things I'll be talking about in detail in the coming slides. So, functionalized scaffolds, where we have the polymer scaffold, and polymer scaffold also you have to choose very wisely, which should have good absorption properties, and you should also curate signaling molecules and growth factors and all these components which are to be there, which are to be there, which have to be incorporated to aid the wound condition. Second is polymer modification. Now, if we always aim for making the system better. So, as when I talk about functionalized scaffolds, you can imagine putting a lot of things in the scaffold. Okay, so if we do not want that, you can always have polymer modification and have the polymer with the intrinsic property. Suppose if you want to have a scaffold which has to release nitric oxide. So instead of putting some material from outside, you can functionalize the polymer of which the scaffold is to be made, and have an intrinsic property of releasing nitric oxide. And third one is the advanced fabrication techniques. Of course, through classical techniques, it is very difficult to reproduce the same size and same architecture of the scaffold again and again. So you need to have some automated system. Also, we want to mimic the tissue in hand. In my case, skin tissue. We want to mimic the native tissue, so we need to have some precision. For that, advanced 3D printing technologies are there, where we can easily print and different type of scaffolds and functionalize them and use them for wound healing applications. So coming to my first approach, that is functionalized scaffolds. Here we have taken chitosan gelatin cryogel. Now, why do I talk about cryogel? Because cryogel automatically gives a very huge advance over hydrogels. They are highly porous. Cryogels are generally, made in sub-zero temperature, and they are highly porous. Easily, if you choose the material wisely, they are easily degradable, and there are different, and since they're highly porous, easy, when vasculature happens and angiogenesis happens, it is very easy for nutrient exchange, oxygen exchange. So all these things are important for scaffold formation. So we take cryogel as our base material, uh, due to its advanced properties. Now, in this work, we have incorporated this scaffold was made up for chronic wounds, chronic infected wounds. So we have chitosan gelatin cryogel. We have an iodine layer, which is made up of PBP iodine complex for bacterial manifestation, since we are talking about infected wounds. And then we have nitric oxide nanoparticles for signaling properties, cerium oxide microparticles for antioxidant properties, uh, calcium peroxide microparticles for oxygen release. This scaffold was also used for under hypoxic conditions. So all these properties we have incorporated in the scaffold. So this is generally an overview of how the scaffolds were made at sub-zero temperature, and some of the characterization results. So few things are very important to keep in mind while you're working and you're developing the scaffold. First of all, you can see the scaffolds, all the groups are highly porous. So that is a very good advantage if you're developing a biomaterial. Second is, you can see water uptake, swelling ratio, degradation rate. So whenever there is a wound condition, there's a lot of exude coming out. So we do not want the wound to be always in that condition. So the cryogel has good water absorption property. So water uptake and swelling ratio depict that. Again, the degradation rate. Now, as I mentioned earlier, a chronic wound normally can take up to 3 to 4 weeks to get healed. So we do not want the polymer, scaffold to be there while the wound is nearing the healing stage. So the degradation property of the scaffold should be parallel with the healing capacity of the wound. So in here you can see in nearly 15 days, more than 90% degradation has happened, and scaffolds will go from there. They'll not be there when the wound is healing. So now all these are the release profiles, the materials and signaling factors which we incorporated showed slow and sustained release over a period of 1 week. Now, another thing very important before going to the in vivo validation and thinking about product development and thinking about clinical translation, one basic thing to be checked with bioengineered scaffolds and tissue engineering approaches is that you should always check for biocompatibility, whether your scaffold is biocompatible or not. So we do that in vitro with cells. Since I'm working on wound healing conditions, I've taken fibroblast cells. These are NIH33 cells. So this is a simple live dead assay which shows, here you can see the dead cells which are depicted by red, and the green cells are the viable cells. So you can see negligible amount of red cells are available. We just grew the cells with conditioned media from different functionalized scaffolds, and all the groups had a good number of viable cells. So that shows biocompatibility. That means your material is not toxic to the cells, and you can easily proceed. Second is, since we incorporated the antioxidant properties, so we also wanted to check that in vitro. So we have made an H2O2 facilitated oxidative cell stress model. So this is the negative control where we have not given any treatment, and the more amount of red cells depict cell death, and where intervention was given and functionalized scaffold was given, the number of dead cells are less. So this can be considered as a main group where almost all the components are present. These are some of the quantifications associated with these tests. Again, scratch assay is very important for wound healing materials. So scratch assay gives you an estimate of how the material will behave when you go for in vivo test. So here we create a layer of the cells. We do it in vitro and we create a scratch in between. You can see. And then we supply the cells with the conditioned media from different scaffolds. So the after 24 hours and 48 hours, we monitor the infiltration of the cells, and you can see in the main group the wound has almost closed in this particular group. So that shows the wound healing potential of our scaffold. These are some of the antibacterial test, time kill assay, and zone of inhibition assay, and all these both the assays showed good antibacterial properties of the scaffold. Now coming on to the main study, which we think about before taking it to the proceeding for the product development stage. This is the in vivo validation. So we try to mimic what is there in the what is the clinical need, and how should we mimic that in our animal models. So we have taken Vistar rats for this study. We have created infection wound model, and we have placed our scaffolds from different groups. So by the end of third week, you can see good wound healing capacity in our scaffold. This one is our main group bilayer, and this is the quantification for wound closure. Second, we also want to see the ultrastructure. As I talked about earlier, that we are aiming for regeneration. We want the tissue to be as closely similar to the original tissue which was there before the trauma. So if we look at the ultrastructure through H&E staining, through mesenchyme, you can see in our main group there's good blood vessel formation, epidermis, this dermis and hypodermis is there, and it is similar to that of our control group. So we will not consider this control group over here because this is negative control, but we can see good skin ultrastructure in our bilayer group. Similarly, collagen formation is there in the mesenchyme staining, and this one is CD31 immunostaining. This is a marker for angiogenesis, which shows good angiogenesis potential of our scaffolds, during the healing conditions. So this paper was published in Chemical Engineering Journal in 23. Now this was the first approach which I took. Second one is the polymer modification, and third is 3D printing. So here what we did in the polymer modification, since I was working with signaling molecules, I had nitric oxide and antioxidative properties, which I understood to be important for healing conditions. So what I did, we did this work in collaboration with a chemical engineer. So we took the gelatin. We wanted to modify the gelatin, chain with something which gives antioxidant property also, and also nitric oxide releasing property. So we modified the gelatin first with N-acetylcysteine, which has thiol group, which gives antioxidant properties, and secondly with the same thiol group, we did the reaction with sodium nitrite, and SNO group was added, which gave nitric oxide releasing properties. So this paper came in. I'll be talking in detail more about in the coming slides, but this particular work came in European Polymer Journal. And so first of all, let me, yeah. So these are some of the results of this polymer modification study, some characterization results, and how we carried out the reaction. One thing I would like to emphasize that since I am a bioengineer, I do not know much about chemistry. So it is very important to collaborate with the appropriate people, with people who can help you with your work, and if you have any idea. So we collaborated with a chemical engineer, and he was very good, and he helped us with this modification thing and all. Second, we again, since we made the polymers and modified it, we wanted to check the biocompatibility, and all the polymers, all the groups showed good biocompatibility. and up to a certain concentration they were fine, but obviously if we increase the concentration way too much, then they are going to be toxic. So we have to choose appropriate and um, uh, appropriate concentration to further work with the polymers. These are some of the functionalization studies where you can see neurite extension, which is the property of nitric oxide, which we did with N2A cells. And these are two of my collaborative works. So through this, I just want to tell you that people do work on these lines, polymer modification, and then further go for scaffold formation. So this work was done in collaboration with Aalto University, Finland. This is Dr. Isabella. So their group was modifying the chitosan backbone and incorporating some antibacterial properties in it. And this is Dr. Rakhi from Department of Chemistry, IIT Kanpur. So her work was on modifying some pyridine bases to get nitric oxide releasing properties. So these are some of the collaborative works. Now coming on to the 3D printing work, we wanted some advanced fabrication technologies. So we introduced 3D printing for this, since it is very precise and you can have reproducible and very precise scaffolds out of it. So this work was also done in collaboration with the Aalto University, Finland, and they had BioX extrusion based printer. So for this work, we took gelatin and cellulose nanocrystals. Cellulose nanocrystals were taken to give little strength to the scaffold. Since in 3D printing it is important that the bio ink which you are preparing should not collapse on each other and the layer should be maintained. So you have to think about those properties and what material can be incorporated to have a sustained scaffold which can be applied at your concerned trauma or whatever is the wound case. So these are some of the characterization studies. Again, it shows some porous structures, and here again porosity and degradation properties are there. So these are some of the biocompatibility studies which we did with the scaffolds. Now this was only plain scaffold. These are some of the concentrations which we took of gelatin and cellulose nanocrystals. What we did, we functionalized these polymers, these scaffolds, with the functionalized polymer, which I talked about in my previous slides, the functionalized gelatin, which has nitric oxide releasing property. So that way you can enhance your work and advance your work. You make something, you advance it and use it in your next work. So that way a continuity is there, and you will always have an upgrade of your material. So these are some of the biocompatibility studies, scratch assay, again neurite extension. So this work came in International Journal of Biological Molecules, Macromolecules in 24, I guess. So again, we wanted to take up a challenge and up the notch with diabetic wounds. So I think everyone is aware that diabetic wounds are very notorious. The condition is like at the wound site, not only at the wound site, because the body's physiology is so much disturbed that the wound doesn't get healed, and it has many conditions at the wound site. One of the main things which we were targeting was amputation, because during diabetic conditions what happens is and since now India is the diabetic capital of the world, so when the skin gets infected, normally SRES is the main microorganism responsible in these kind of infections. So once the cut is there and some small wound is there, and the wound gets infected with SRES, and if it is not treated and not taken proper care of, it can penetrate and it can reach up till the bone, and it can manifest there, and which leads to amputation. So this was something we wanted to target in our next work, and this work is dedicated to that. So what we decided, that we will have a dual strategy. So this Kragel scaffold we made for skin, for the upper wound which is there, and again antibiotic loaded bone cement. This is nanohydroxyapatite based bone cement loaded with rifampicin, which is a broad spectrum antibiotic. So we use both these scaffolds. So skin scaffold was for the skin wound, and antibiotic loaded bone cement we inserted it in the bone infection. And again, in this skin scaffold, I use my functionalized gelatin, which I showed earlier, which had nitric oxide and antioxidant properties. So for antibiotic, we have rifampicin, and for enhanced therapeutics, since many of you must be aware about the extracellular vesicles and exosomes are of great therapeutic value. We also used incorporated VMSC derived exosomes in our scaffolds. So these are the scaffolds, skin scaffold and bone scaffold. These are again some of the characterization studies. All our scaffolds are biocompatible and safe to use. This work is currently under revision in Material Chemistry B journal. So yeah, this is the main thing which I wanted to emphasize upon, the model development. Whenever you are taking up a problem, you should always think that what is the clinical scenario, how the patients are there, and what is the condition there, and try to mimic that in vivo. So here what we did, we again worked on Vistar rats here, and we made a around 8 mm wound on rat tibia, and in the same line we punctured the bone in the tibia. We made a small 1 or 2 mm hole and infected it with S aureus. So both upper skin wound and bone was infected with S aureus, and after the bacterial manifestation and this uh, condition was developed, infection condition was developed, we cleaned the wound and placed the scaffold. This is the bone scaffold which we are inserting in the bone defect, and this is the skin scaffold. And after healing, it looks something like this. So we monitored the wounds through 4 weeks, through a total time of 8 weeks. So you can see in the negative control how bad is the condition. You can even see the bone is visible, and skin has almost disappeared. But in our main group, that is where both skin and bone was treated, both these groups. And the fourth group was also we incorporated exosomes. So both these groups had good healing properties. So this is ultrastructure of the skin after the sacrifice and bone formation after 8 weeks. Some of the histology results. So up till now, I talked about my work and what strategies I took. But most important question and most important insight which I would like to suggest to the audience attending the talk today is that most of us, I was also in that stage, so most of us are limited up to the lab stage. We have a good idea, we know how to carry out all the experiments, and we know the in vitro models and in vivo models. In my case, since I'm into biomedical engineering, so all these terms are familiar to me, and all those who are working in the area may also relate to it. So but what should I do after that? I have this prototype, I have this scaffold. Now what should I do? So these are some of the terms and some of the lines which you should be aware about, and you should be taking notes of that, how to advance your research from lab to market for use of the people, for clinical use, to help mankind. So TRL stages are very important. TRL stands for technology readiness level. So this goes from TRL 1 to TRL 9, where TRL 1 is the generation of idea, and TRL 9 is when your product has finally reached the market. So we are good up till here. Whatever I talked about in my previous slides was up till here, TRL 4. So everyone is very happy till TRL 4, getting all the publications and this. But what happens really happens if you want to advance your product after that. So TRL 4 to 7 is the value of death, as we call it. So most of the researchers are clueless, and in spite of having very good products, very good innovative ideas which can be translated into good products which will be very helpful, they are not aware about many things. So what happens after TRL 4 comes TRL 5, where first of all, it is very important to protect your technology. So whatever material you are developing, whatever uh, prototype is there, first of all file a patent. IP protection is very important. So you can have patent filing can be there, copyrights and different things. So it should not be the case that you're working on something, someone hears about it, you do not have the patent for that, and someone else does the work, and he has the right connection, so he gets ahead of it. So you should always protect your technology. IP protection is for that. So in DRL 5 you file for protection and file for different regulatory clearances also. So here is when, for especially for medical products, CDSCO comes in, which is the main regulatory body in India. So you file for test licenses, manufacturing licenses, and all these things are there. So all the data which you have collected, they will also ask you for more data in spite of having all the lab generated data. But you collect all the data and submit it to them, submit an application for test license. Test license gives you a clearance of conducting clinical trials, or clinical trials of your product. So once you get that clearance, because you cannot develop a medical product without going into clinical trial, right? For that also there are different categories. Like some products have a predicate means some of the earlier products are on similar lines and clinical trials have been developed for them. If your new product is like innovative and is on similar lines, then clinical trials can be skipped. But in most of the cases, if the product is new, clinical trials are needed. So for especially for those where the predicate is not available, you have to go for clinical trial, where you will need all the clearances. And again, once the clinical trials are successful, you go for manufacturing. Obviously, since we are researchers, we are working in lab, we do not have manufacturing facility. So this is where you connect with the right people, connect with the right industry which is working in the area, collaborate with them, show your product, and if they are interested, they will manufacture your technology. And again, for product launch and marketing, you can again collaborate with the company. Some of us might also put up a manufacturing facility and go for marketing ourselves, but it is a huge task, I think. And most of us are like oriented towards research and more towards R&D. So these are some of the things which you should for which you should have the right people, I think. So it is very important to collaborate with the correct people if you want your product smoothly to come into the market. And something which I already talked about, that our country is very good. It has very good research ideas, but if there are 100 good research ideas, there will be 30 working prototypes. If prototypes are there, then animal studies are so limited, and there are so many clearances also. So 10 will go into animal studies, 3 will go into clinical trial, and only one commercial product will be formed. So out of 100 research ideas, you have one commercial product. And what is the reason? Lack of real clinical need. Yes, this factor is very important. You should always think about problems which are of mass value. If you take up something, some disease, although that is also important, but if you take up something which is only valuable to 0.01% of the population, then to whom you are going to sell the product, right? So if you have idea towards something which will be beneficial for many people, for most of the population, it will be very good. Your product will be a hit. So poor scalability and reproducibility, again, lack of manufacturing facilities and lack of connection, no intellectual property protection, regulatory hurdles, manufacturing challenges, lack of industry partnership, and funding gap. So these are some of the challenges which people working in translational research do face. So these are some of the points which, if you are really targeting for market and want to launch your product for clinical use, then these are some of the lines which you should really look into. And since I am associated with this Varvani Innovation Network Center of Excellence, this center is purely dedicated towards translational research. So we try to help projects which are really at an advanced stage. So we do not take projects, we do fund, and in various collaborations also we do help the project, but we do not take the projects at a very advanced stage. We take the projects which are at late TRL 4, where the prototype has already been developed, but the researchers and the team is really lacking the connections and clueless about what happens next. So this is where we come in. We fund the product and fund the project, and try to help the project in all the ways possible to bring it into the market. So some of our strategies to support the project. Yeah, so I would like to close my talk with this, and some of these are my mentors, Professor Ashok Kumar and Professor Kavita Shah. Some of our international and national collaborators, funding agencies, I want to acknowledge, and thank you for all for listening and having patience. And if there are any further discussions, if you want any help and support for your project, and you want to put up a project in the Center of Excellence, you can always contact me at this email ID.
Thank you so much, ma'am, for your insightful lecture. Participant, you can unmute yourself, or you can raise your hand to ask any query if you have, or you can write in the chat box also. If you have any query, we will take your question one by one. One question is there, ma'am. If we publish research work in journal, then can we go for the patent filing?
Sorry, I didn't get that.
Ma'am, there is a question in chat box. They
Yes.
want to ask that if they have published their paper in a journal, they
Yes.
can go for the patent or not?
No, no. You have to go for the patent before the publication happens. So this is very important. So you should always first file the patent and then go for publications. Once you have that application number of your patent, then you go for publication. Till then you wait. If your product has that potential to go into the market.
Okay. Any other question? If you have, please raise your hand. I will unmute you. Any participant, if you want. Uh, One another question
At what
asked by.
stage of patent we can go for publication, right? This is the question, I think.
Yes, yes.
So once you have filed the patent, you have the application number, so then you can proceed for publication.
Rupita ma'am, are you here? Hello, Rupita ma'am, are you here? So we are waiting for 1 minute. If any participant want to ask any question.
I think I finished before time.
Okay.
Yes, phage therapy is another very interesting area, and we also have someone has asked in the chat box, I think, about phage therapy. So it is very effective, and you can always have it a separate as a separate therapy also, or you can combine it with the biomaterial. So those are also some of the areas where our group is working.
So I think there is no question. What is this? What about the phase therapy, okay, for wound healing. You have addressed, ma'am. Artificial intelligence in antimicrobial resistance, drug repurposing. What you want to say? Uh, Santosh Krishna Chaitanya. Can you rephrase your question? Or please raise your hand. I will unmute you. You are not audible. Chaitanya, Santosh.
So you can always write to me or connect me on
Yes,
LinkedIn
yes, yes,
to
yes.
have an elaborate discussion.
Yes, ma'am. Okay, so once again, thank you so much, ma'am, for accepting our invitation, and I am thankful to Rupita Ghosh, ma'am, also to for connecting you with us with the Department of Biotechnology at Calcutta University. So your presentation was very informative, and
Thank you, sir, and I truly appreciate all of you and the organizing team for having me here and inviting me for this talk. It is always great to connect with young minds and give them some food for thought if they are working on these lines.
yes, ma'am. So I am also working on the wound model, and
Okay, okay.
yes,
So
so
we can talk
in the,
more after the session, after this
yeah,
workshop
yes,
and
ma'am.
program
So
is over.
previously we have published some paper on the scaffold. We have developed a nanocomposite and some scaffold to check the wound healing property in diabetic wound and chronic wound. It was very interesting that to see that you are also working on this. I have also submitted some project on the wound dressing in the big grant, but unfortunately I didn't get it.
Okay.
Okay, so
So there are many funding agency. You can also, if your project is at an advanced stage, you can always come to us. We'll
yeah,
be opening calls again, I think in November, December. So you can submit your proposal there also.
okay, okay. So, ma'am, on behalf of the Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Calcutta University, and our collaborator, University of Cyber Jaya, Malaysia, I am honored to offer you this digital memento
Thank you
for
so much.
delivering such an informative and wonderful session, an interactive session. Thank you, ma'am, and kindly accept it, ma'am.
Thank you so much.
Thank you. And participant, I have sent, posted the feedback form in the chat box. So kindly provide your feedback. Um, and I am also grateful to all the participants that you have joined. You have spared your some time from your busy schedule, and I think this is our overwhelming response we have got for this faculty development program, and we got approximately 1400 registration, and approximately more than 250 participants per day are attending. So thank you all the participants for making this event grand success. Thank you everyone. Thank you, ma'am. Once again, thank you, ma'am.
Thank you. Can I log off now?
Yes, yes, ma'am.
Yeah, yeah.